Do you know how many types of biomolecules can be used in a biosensor for sensing of a target? Let's discuss that.
These are the biological molecules which are used for detection of analyte in a biosensor. They can be an antibody, an aptamer, a whole cell, an enzyme, a single stranded DNA, etc. Biomolecules use is not new for detection. We have been using them in techniques like ELISA, enzyme linked immunosorbent assay, in which antibodies are used in different configurations for detection of the analyte. So, the idea behind ELISA and biosensors is same, that we are going to utilize different biomolecules for detection of the analyte. In ELISA, only antibodies were used for detection of the antigen, but in the case of biosensors, you have different types of biomolecules available.
To know more about biosensors, stay tuned.